Welcome back to the University of Melbourne Biology series. This is episode 3. So far we have built life from atoms up to the 4 great families of molecules. Today we put one of those families to work. Picture a cell as a tiny city. It needs a wall to mark where it ends and the outside begins. It needs gates in that wall to choose what comes in and what goes out. And inside, it needs roads, factories, and recycling centers. Over the next 12 minutes we will build all 3. We start with the wall itself, the membrane. Then we open its gates and watch substances cross. And finally we step inside to tour the cell's internal network of compartments. Let us begin at the boundary. The wall of the cell is built from a single clever molecule, the phospholipid. Every phospholipid has 2 halves with opposite tastes for water. At one end sits a hydrophilic phosphate head, a part that interacts happily with water. At the other end hang hydrophobic fatty acid tails, which are repelled by water and instead are attracted to one another. So a single phospholipid cannot decide whether it loves water or hates it. It does both at once, at opposite ends. Keep both of those facts in mind, because each one is about to matter. The heads will face the water, and the tails will hide from it. That split personality is not a flaw. As we are about to see, it is exactly the property that lets these molecules organize themselves into the boundary of every cell, with no outside instruction required. Now drop a crowd of these phospholipids into water and watch what happens. The hydrophilic heads turn to face the water, while the hydrophobic tails turn away from it and huddle together. The result is 2 sheets of phospholipids back to back, a phospholipid bilayer, with the tails sandwiched in the middle and the heads facing the watery worlds on either side. This bilayer is the plasma membrane. And because the oily core in the middle repels water, the membrane prevents the free flow of liquids into and out of the cell. In other words, the very same water-fearing tails that drive the membrane to assemble also seal it, turning a loose crowd of molecules into a controlled barrier around the living cell. Here is a subtle but vital point: the membrane is not a rigid solid. It is fluid, and it has to be. The phospholipids that make it up are constantly moving, and that movement is what keeps the membrane fluid. This fluidity is crucial for the membrane to function properly. A frozen, rigid membrane could not do its job. But fluidity raises an obvious danger. If the membrane simply got more fluid as things warmed up and stiffer as they cooled, the cell would be at the mercy of temperature. The cell needs a way to steady the membrane across a range of conditions, to keep it fluid enough but never too leaky. Remarkably, it solves this with one extra molecule tucked among the phospholipids. That extra molecule is cholesterol. Cholesterol is a nonpolar molecule that sits among the phospholipids and forms hydrogen bonds with the adjacent fatty acid tails. Its real talent is acting like a thermostat for the membrane, and it works in 2 opposite directions. At high temperatures, when the phospholipids would otherwise spread apart and the membrane would leak, cholesterol enhances the packing of the fatty acid tails, holding them together and reducing permeability. At low temperatures, when the phospholipids would otherwise crowd together and solidify, cholesterol disrupts that packing, increasing permeability and keeping the membrane workably fluid. One molecule, 2 opposite jobs, both aimed at the same goal: a membrane that stays just fluid enough whatever the temperature. We have our wall, and we know it is sealed against the free flow of liquids. So how does anything get across? There are 2 ways, and the cheaper one is passive transport. In passive transport, substances move down their concentration gradients, from where they are crowded to where they are sparse, and the cell spends no energy at all. The simplest form is diffusion, the ordinary movement of a substance with its concentration gradient. A special case is osmosis, which is the diffusion of water across a selectively permeable membrane. And when a substance is hydrophilic and cannot slip through the oily core on its own, the cell offers facilitated diffusion, which uses transport proteins to let those water-loving substances pass. All of this is downhill, and downhill is free. But cells often need to do the opposite of downhill. They need to move a substance against its concentration gradient, from where it is sparse to where it is already crowded. That is active transport, and pushing uphill is never free. Active transport requires energy. There are two ways to pay the bill. Primary active transport uses ATP directly to move molecules, spending the cell's energy currency right at the pump. Secondary active transport is more cunning. Instead of burning ATP itself, it uses an electrochemical gradient of a different substance to drive the transport of the target molecule, riding one substance's stored gradient to push another uphill. Passive transport coasts downhill for free, while active transport pays, directly or indirectly, to climb. We have built the wall and opened its gates. Now let us step inside, into the cell's internal network of membranes, the endomembrane system. This is a connected set of membrane-bound compartments, and it has a clear membership. It includes the nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, the lysosomes, and, in plants, the plant vacuoles. Working together, these compartments carry out one grand job: they synthesize, package, and distribute the cell's macromolecules, its proteins, lipids, and carbohydrates. Think of it as the city's combined factory, postal service, and delivery network, all wrapped in membrane. Over the next few minutes we will visit its main stations in the order a product would travel, starting where most of the building happens. The first station is the endoplasmic reticulum, a sprawling network of membrane-bound compartments that serves as the factory floor. It comes in two versions, and the simplest way to tell them apart is to look at their surface. The rough endoplasmic reticulum is studded with ribosomes, and it is involved in protein synthesis, which is exactly what those ribosomes are for. The smooth endoplasmic reticulum lacks ribosomes, and instead it is involved in lipid synthesis. So one division builds proteins while the other builds lipids, two product lines running on the same factory floor, and both feed their products into the same delivery network. Whatever the endoplasmic reticulum makes does not stay there. It is loaded up and shipped onward to the next station for finishing and sorting. That next station is the Golgi apparatus, the cell's packaging and dispatch center. Structurally it is a set of flattened stacks of membrane called cisternae. Its role is to collect, package, and distribute the molecules that were synthesized in the endoplasmic reticulum. And it runs as a one-way line with a clear direction. Vesicles arriving from the endoplasmic reticulum enter at one side, called the cis face. The molecules move through the stack, and finished products leave from the opposite side, the trans face. So cis is the receiving dock and trans is the shipping dock. By the time a product leaves the trans face, it has been packaged and is ready to be sent to wherever in the cell, or beyond it, that it needs to go. Not everything in a cell is being built, some of it is being broken down, and that is the job of the lysosomes. Lysosomes are single membrane-bound organelles filled with hydrolytic enzymes, the molecular tools that digest things, and they keep an acidic interior in which those enzymes work. They break down materials and recycle the cell's old organelles, acting as its recycling center. Plants run a close cousin of this organelle, the plant vacuole. Like a lysosome, a plant vacuole is a single membrane-bound organelle and it too contains hydrolytic enzymes, so it can serve as a degradative compartment in the same way. But the plant vacuole does more besides. It also stores nutrients and pigments, and it maintains the cell's turgor pressure, the internal push that helps keep a plant cell firm. So one organelle has been turned into a multipurpose store, recycler, and support. There is one last trick the cell needs, a way to move things that are far too big to slip through a protein or diffuse across the membrane. For these the cell wraps cargo in membrane and moves whole packages across its boundary. Bringing material in is called endocytosis, the process of packing materials into a vesicle for transport into the cell. Sending material out is called exocytosis, the release of materials and waste out of the cell. With these, the loop closes. The endoplasmic reticulum builds, the Golgi packages, vesicles deliver, lysosomes recycle, and endocytosis and exocytosis carry whole bundles across the very membrane we started with. The wall, its gates, and its inner network, all one connected system. That is the cell as a working city. See you next time.